everyone, I'm Jessica VASU. My matrix number is 23DBT 20F 2019 and I'm from Polytechnic Nilai Negeri Sembilan. I'm here today to perform the virtual bacterial identification for my DYB 30093 molecular biology subject. There are 4 basic steps involved in bacterial identification. First is to prepare a sample from a patient and isolate whole bacterial DNA. Second, make many copies of the desired piece of DNA. Third, sequence the DNA. And lastly, analyze the sequence and identify the bacteria. So let's get started. The first part is sample preparation. To begin, let's put on a pair of rubber gloves by clicking the glove box in the open drawer. Now we will obtain the sample from the culture dish. Click the wire loop to continue. Click on one of the bacterial colonies in the dish. Click the microcentrifuge tube to transfer the bacterial colony from the wire loop into the microcentrifuge tube. Now we will add digestive enzymes to our sample. Click on the yellow micropipette to continue. The microcentrifuge tube must sit for several hours. Let's click on the microcentrifuge tube to continue. After several hours of enzymatic digestion, our sample is ready. Click the microcentrifuge tube to heat inactivate the digestive enzymes. Now we are moving the centrifuge to the microcentrifuge. The digestive enzymes are now denaturated and inactivated. Click the microcentrifuge tube to continue. Now we will spin down cellular debris for removal from our sample. Click the centrifuge lid to continue. Centrifugation is complete. The unwanted debris has now been spun down. DNA we want is in the supernatant. Click the microcentrifuge tube to continue. The sample preparation is complete. Click the orange tree to continue to part 2, which is PCR amplification. We will now add the Pcr master mix solution to all tubes. Let's click on the red cap master mix bottle to continue. Now we need to set up the positive control reaction. Let's click the green capped positive control DNA bottle to continue. For the negative control reaction, we will add water. Click the blue capped deionized bottle to continue. Click the PCR machine to open the loading compartment and load the PCR tubes. Now we are loading the PCR tubes. The PCR tubes has successfully been loaded into the PCR machine. Click the PCR machine to continue. Running Pcr amplification. We are on cycle 1. We have to go through 30 cycles to complete the PCR. Now the PCR amplification is complete. Let's click done. The PCR amplification is complete. Click the orange tray to continue to the PCR product purification process. First, we will need to set up the micro concentrator column. Click the yellow cap buffer bottle to continue. Now let's click the orange tray to continue. The tubes containing the PCR sample positive control and negative control will be put on ice. Click the orange tray to continue. Click the orange tray to load the column into the centrifuge. Loading the column into the centrifuge. Make sure to add the counterbalance and click the centrifuge to continue. DNA is now trapped in the column. Now we need to loosen it into a new tube. Click the orange tray to continue. Click the inverted column to continue. We are adding the buffer solution to the inverted column. Make sure to discard the tape after each process. The first collection tube is no longer needed. Click the orange tray to discard it. Now we will centrifuge to assembly to collect the DNA into a collection tube. Let's click on the orange tray to continue. Loading the column into the centrifuge and add counterbalance. Let's click the centrifuge to continue. DNA has now been collected into the collection tube. The column is no longer needed and will be discarded. Click the orange tray. The PCR product has been purified. Click the orange tray to continue to part 4, which is DNA sequencing. First, we will dilute the PCR product to increase the volume. Click the blue cap distilled water bottle to continue. The green and blue striped tubes already contain PCR reaction mixtures in them. Click the strip tubes to continue. The strip tubes are ready for PCR amplification. Click the PCR machine to continue. Now we are loading the strip tubes into the PCR machine. The strip tubes has been loaded. Click the PCR machine to continue. This is the PCR amplification results. Done. The sequencing preparation is complete. Click the orange tray to continue to part 5, which is DNA sequencing. Now, we will load the automatic sequencer. Click the orange tray to continue. Click the DNA sequencing machine to open the doors and load the tray. Click the orange tray to load it into the DNA sequencing machine. Click the inner grey door above the orange tray to view the DNA sequencing animation. Now the DNA has been sequencing.
I ate and left no crumb.
Now the sequencing is finished. We have completed the DNA sequencing section. Click the orange tray to continue to part 6, which is sequencing analysis. Follow the instructions in the notebook window. Click the computer screen when you're ready to identify the bacteria. For DNA sequencing analysis, we are working with sample A, which is a fluid from lymph node. See sample section for more info. Let's go to the sample section. Sample A was obtained from tissues and fluid aspirated from a lymph node of a patient complaining of a fever, fatigue, and swollen lip glands. So let's go back to the notebook section. Now, let's follow these steps to identify our sample. Open the data output from the sequencer. This will open a new browser window. Select and copy the sequence data inside the gray box. Go to the NCBI website to perform our search. On the NCBI website's splash page, under the Enter Query Sequence section, paste your sequence data from step 1 into the box labeled Enter Accession Number. Under the Choose Search Set Database section, standard databases and nucleotide collection should already be selected. When we are ready, click on the blue blast button further down the page. So let's go to the data sequencer, data output from the sequencer first. So this is our data. Let's copy it. Now let's go to the NCBI site to perform our search. This is the NCBI section, which stands for National Center of Biotechnology Information. On Enter Query Sequence, let's paste it. On the database section, standard databases and nucleotide collection has already been selected. So now let's click the blast button and wait for the results. So we are still waiting for the results. Okay, here is the blast search result. So according to the result, we have Bartonella henselae and some other uncultured bacterium clone identified in this sample. Let's go back to the bacterial identification. The bacteria is Bartonella henselae. Yes, we have identified the bacteria correctly. Now, let's perform DNA sequence analysis on other samples. Next, we are going to go to stool sample. This sample was obtained from the stool of a patient with a normal temperature complaining of several abdominal cramps and watery diarrhea. Now let's go to the data output from the sequencer. Basically, we are going to just repeat this process until all the samples are clear. So as we see, the standard database and nucleotide collection has already been selected. Now let's click the blast for result. So the results are here. Let's go down. So according to the result, Escherichia coli is the main bacteria identified in this sample. Let's go back to our lab simulation. So the bacteria would be Escherichia coli. Now let's perform analysis on other samples too. Sample C would be urine sample which was obtained from the urine of a patient complaining of pain during urination and a discharge from the urethra. We are going to repeat the same process over and over until we identify all the bacteria in the samples. The results of the urine sample is here. According to the results, Dominus aeruginosa is the bacteria that has been identified with the person identification of 100%. So let's go back to our simulation. So the bacteria would be Pseudomonas aeruginosa. Now let's go to sample D, which is a blood sample obtained from a blood of a patient complaining of fever, headache, joint pain, constipation, loss of appetite, and abdominal pain. According to the result, Salmonella enterica is the bacteria identified in this sample. So the bacteria would be Salmonella typhimurium. Let's go to sample D. Sample E would be the sputum sample, which was obtained from the sputum of a patient complaining of chills, high fever and cough with labored breathing and sputum with fleck of blood. Oh my god. Let's copy the data sequence. So the results are here. According to the result, Yersinia pestis is the bacteria identified in this sample. So let's go back to our lab simulation, and the bacteria would be Yersinia pestis. Now let's go to our final sample F, which is a stool sample obtained from a stool of a child who has fever, abdominal pain and bloody diarrhea. Let's paste it and blast. So the final bacteria identified in this last sample would be Yersinia enterocolitica. Let's get back to our lab simulation, and the bacteria would be Yersinia enterocolitica. So all the samples has been identified correctly, and that's all from me today. Thank you for watching. Bye.